One of the more interesting topics we covered this week was how the ear worked. I had never really given any thought into how our ear translates sound for our brain to understand. But after learning about it, I found it very interesting. So in this video, I want to cover how the ear does this amazing feat and how we understand and process the sounds we hear around us. But first, we must start with the simple explanation of sound and how it works. The sounds we hear all around us every day, from the chirping of birds to police sirens to even dog barks, are all just vibrations of molecules all around us. When a speaker makes a sound, it will push all the nearest molecules forward, transferring this motion to the next molecules in a wave-like fashion until they reach a wall or our ears or something else. This wave-like form is what we call longitudinal waves. To better understand these longitudinal sound waves, we represent them using transverse waves. Using this transverse model, it is easier for us to understand how the amplitude and wavelength correlate to the pitch and loudness of a certain sound. As we can see, shorter wavelengths correlate to higher pitches, and longer wavelengths correlate to lower pitches. This would be represented in a longitudinal wave format by the compressions either being more compressed together or spread out apart further. Now, when looking at loudness, a higher amplitude means that a sound would be louder, while a lower amplitude represents a sound that would be quieter. This would be shown in the longitudinal graph by the molecules, either more molecules being represented in each compression, or with the lower amplitude, less molecules being clumped together in each of those compressions. Now that we have a simple understanding of how sound waves work, let's discuss the main components in the translation process in our ear. The diagram above shows the different parts of the ear that all contribute to this process of translation. As we can see in red, there's the eardrum. This thin skin-like membrane works like the head of a speaker, whereas it vibrates to the sounds entering the ear. Then, using the bones connected to the cochlea, it will translate those sounds that it hears in the ear into vibrations, where the bones will tap on the cochlea, representing those sounds that we just heard. Now, once those vibrations enter the cochlea, they travel down the tube all the way to the end. As we can see, the cochlea is a seashell-shaped organ inside the ear, and as we can see in the diagram, there are little hairs that can sense when vibrations pass over them inside the cochlea. Now, these hair cells are very important into this process. As they shift and sway with the vibrations inside the cochlea, they release ions into the hair cell, where they will translate into neurotransmitters that's sent down to the auditory nerve. And as the hair sways, the more it sways, the more ions are released into the hair cell. This is how the brain is able to tell how loud a sound is, as the more the hair spindle is pushed, the more ions are pushed into the hair cell. Going back to the previous diagram, as we can see, with the cochlea in a seashell shape and it thinning out towards the end, this is how the brain tells the differences in the frequencies that we hear. The reason that the brain can tell the difference in frequencies is based on the shape of the cochlea. As we can see in this diagram, it thins out towards the end, and as we learned from the sound section, where higher frequency sounds are represented by shorter wavelengths, the lower wavelengths are the only ones that can reach the farthest section in the cochlea. And this, based on which hair cells trigger all the way towards the end, is how the brain is able to tell the differences in frequency.